Welcome to Birch. My name is Brian Frstensky. Today's video is an update on the latest developments in Birch, a comprehensive system for bioinformatics. And I want to start with a quote from a commercial a long time ago. At BASF, we don't make a lot of the products you buy, we make a lot of the products you buy better. Now, to me, this conveys perfectly the central ideas behind Birch. Just having a lot of programs installed on your system is only half the battle won. How do you know which ones are available on your system? Where's the documentation? How much time will it take you to figure out how to format output files from one program to be used as input for another? How long will it take to figure out the quirks of running each program? So this is the problem we're trying to solve with Birch. And the way we do that is through a family of applications known as BioLegato, each of which is specialized for a particular type of data. For DNA sequences, there's BLDNA that works specifically with DNA sequences, BLProtein for protein sequences, BLAnalign and P-align for DNA and protein alignments, BLreads for next generation sequencing reads. So each type of data is packaged along with the programs that work specifically on that type of data. And so if you know anything about programming, this would be very similar to the same concepts in object-oriented programming. Today I'll illustrate the new functionalities in BIRCH 3.8. If you want to skip ahead, the start time for each topic is listed. Remember, the unifying theme is showing how BioLegato makes the programs better by making it easier to run them, to go from one step to the next, and to experiment with your data, a process we call ad hoc pipelining. Ideally, a sequencing project should start with raw sequencing reads, and then usually you'll trim the reads with a program like Trim-O-Matic, and then probably do some kind of error correction. If they're RNA-seq reads, for example, you would run rcorrector and then get corrected reads, which you could then use in an assembly program like Trinity or RNA-Spades. But there are often problems with sequencing reads at different steps in the process, and we'll take a look at how we might fix some of those problems. So one of the things you sometimes worry about is whether or not you've got substantial amounts of read through. In other words, short reads where you start reading at the 3' end into the adapter. So I've got a bunch of fastq files here, and I'm going to check them using a program that has just been added in BIRCH 3.8 called Adapter Check. So you can either search previously defined sets of adapters or some custom set of adapters, and then search for all of those adapters in the sequences, and output will go to a spreadsheet. So when adapter check is completed, the spreadsheet pops up, and you can see that for each file we have a report on all of the primers, or I should say adapters, that were checked. And so we have the total number of sequences, and then for that number how many of these adapters should be found. Many of them are actually none are found, but for these two adapters, over 51,000 out of about 2.4 million reads had some kind of read through. So at least we know that these are the adapters we want to search for, and we know that for at least some sequences it's a good idea to get rid of them. So the next step would be to run Trim-O-Matic using this adapter set. So another thing that you can encounter at almost any step in working with sequencing reads is a file that contains some bad reads. This can happen for a number of reasons, but we're looking at a fastq file here, and all reads should be in 4 lines. A name line beginning with an @ sign, the actual sequence, another name line, and then the quality values represented as letters of the alphabet. So if anything doesn't match this formula for a read, it will be removed from the file. And that's done using a program called fixfq.py. And the other thing that this program will do is let you set a minimum read size. So if the read is too short, it will be eliminated from the from the output. So let's run that and see what we get when we run fixfq. So now we get in the output a report on each of the files that was checked. So let's just take a look at the example of the first one, this 18-1R1. So valid reads, all reads appear to have been valid. There are 0 reads going to the bad file. So you'll see on the right here in BL reads, we have the original file and then the 2 files that were written based on that. So in this kind of a case where it looks like all of the bad files had a 0 size, we can just go with the original files. But this is important because sometimes we just get, for whatever reason, a corrupted read in a file. So the ability to remove those is really quite valuable. So now we have trimmed the reads and we've run them through a read corrector and we've got corrected reads. But one of the problems is that during this process of the different steps, sometimes you lose one member of a read pair. And that can sometimes prevent assembly programs from doing their job properly. And so we have the ability to run a program that will check whether both members of the pair are present. So first thing we have to do is actually to pair up the read files. So we're going to search for all fastq files. R1 is going to be the forward read, R2 is the reverse read, and now guess pairs will correctly pair up the 2 files so that we can now work on these in a batch. So we select all of the read pair files and we're going to run a program called fastq pair. And so what fastq pair does is to make sure that all of the reads that can be paired are paired. So I'm going to start this program going and maybe send a message to my mailbox when it's done, and we'll come back when we have the output. So when fastq pair is done, we get a report that tells us for each file the number of left and right reads, and in this case they look good because the singles are all 0, and then it also tells us the files that they're written to. One other thing, by the way, many of these programs will also tell you the elapsed time, and so this is useful because you want to always plan how long is my task going to take. So here are the same files shown in BL reads. The paired are highlighted and you can see that the single ones are 0. So this was a good result. It just shows us that we know all of the reads are properly paired. There shouldn't be any problem now moving on to the actual transcriptome assembly, which is the main point of what we want to do. Now finally, I just want to briefly cover something that is not going to happen for most of you, but this is from an example in which an algal genome was sequenced and a lot of prokaryotic DNA contamination was found. So the reason is that many algae have bacterial symbionts that the DNA will co-purify with the algal DNA, and so when you sequence the genome you get both. So one thing you might want to do is to see if you could eliminate the reads from the original set of reads, so eliminate the prokaryotic sequences. So in order to do that, we need to be able to search for those, and for that we need to have a BLAST database specifically for the prokaryotic genomes that we're worried about in contaminating our sequences. So here is a list of the bacterial contaminant species that were identified. So what I've done then is to make a FASTA file containing the genomes for each of these species. So that's the file listed in the input RNA DNA file in BLDNA. And so that's the file that we're going to use to create a BLAST database. So a BLAST database takes up many files, and when I ran make BLAST DB, the output that I got included the quite the selection of output files that BLAST needs in order to find all of its information for database search. So that's the first step. So now that we have that database to search, we can take any fastq file or set of files for that matter and run Magic Blast against our user-created BLAST database. So here we just select any of the files from the BLAST database, and in this case it will write two files. One file will be sequences that match the contaminants, and others will be sequences that don't match the contaminants. And so when we run this, we'll get output for both possibilities. So the output from Magic Blast will include if we have the original fastq file, we'll get two new files that include so the plus file will be a fastq file that has the ones that matched. So that's the prokaryotic contaminants, and that's a relatively small percentage of the total, but it's a significant percentage. And then the larger file is the minus file, the presumptive eukaryotic algal sequences that didn't match the contaminants. So this would be one way of then we would be able to extract the where we have the reads and just use these reads as the ones that we use for genome assembly when we're trying to assemble our algal genome, and hopefully be able to get a better result from that. So the underlying theme of most of what I've been talking about is that we're making the programs you use better. And we've already had trimming programs and correcting programs and assemblers, but by adding on a variety of programs that help you with the unexpected things that can become a problem, we end up making the entire pipeline a lot better. So this is one of the recurrent themes that we're going to see with the BIRCH system that makes it very powerful for many of the things that you do in bioinformatics. So continuing with our theme for making the programs you use better, I want to take a look at how the BioLegato program BL Protein makes it easier not just to run BLAST searches, but also to get the most out of the data that you work with. And so to start, we're going to go to the database menu, and there's a new item here that will let you generate a report on whatever databases you have currently installed for BLAST. And so you can see here that there are 4 BLAST databases plus the taxonomy database. RefSeq RNA is a very large one, so if we were going to search that, we would want to be able to factor in a fair bit of time, whereas things like Swissprot or PDBAA are relatively small databases, and so those searches are going to go quick. And for that reason, in this demonstration, I'm going to take a look at a Swissprot search. So we start simply by selecting the sequence we want. In this case, I've got a phenylalanine ammonia lyase gene from parsley, and I'm going to look for other PAL genes. This is a defense gene in plants that's involved in activation of the phenylpropanoid pathway. And so if I open the database menu, you can see that I could search either local copies of BLAST databases or remote copies at NCBI. I'm going to search our locally installed copy of Swissprot, and I will launch the search, and then we'll come back when the results are ready. So as you can see, we get results in a number of forms. We get the traditional HTML results that show you the alignments and the names of the hits. And then we get much of the same data in BLP fetch, which has things like the name of the species the sequence comes from, and not surprisingly the parsley sequences are at the top. And we'll get back to that in a little bit, but probably the most exciting one of the applications we have is BlastViewer, and this lets you go through the results in a number of different ways. You can have a table of the results and then a graphical output that shows you, for example, which hits take up the entire length of the protein, and then as we get to less significant hits, we see that they're only overlapping the search or the query protein by in a partial way, and then even a multiple sequence alignment that is includes a sequence logo that gives you an idea of the conservation of amino acids. So based on these kinds of results, I often find the graphic results to be the most useful, and so I might want to choose, let's say, only those that are going to give me more or less a full length output. And so that would be up to this sequence Q68G84, and because I've selected that particular one. And so if I go back to BLP fetch, you can see here that the 58th hit is the one we were talking about. So we would want to have every hit from there going up to the top, and now we can retrieve all of those hits. So I'm going to run seq fetch, and then the output will come up in a BL protein window that lets us work with those top blast hits for further analysis. So to finish up showing you how Birch makes the programs we use better, I want to show how we work with a number of different programs related to phylogenetic analysis. And the starting point, of course, for phylogenetic analysis is a sequence alignment. Now, the most convenient type of alignment to work with is a protein alignment. We see here a set of chitinase sequences from plants. So this is another plant defense protein that have been aligned using MAFT, and so that's really my starting point. But what I want to demonstrate is that we can also work with the corresponding DNA alignment. And the reason you might want to work with the DNA alignment is that if you think about the degeneracy of the genetic code, you know that we lose information when we translate DNA into protein. There are silent mutations that we wouldn't detect because we don't get an amino acid substitution, and even with conserved substitutions that may still underestimate the evolutionary distance, the number of evolutionary events that have occurred. And so in the ideal, it would be nice to be able to work with the DNA alignment, but the problem is it's very hard to align DNA sequences, really impossible in most coding sequences. So the best way to get around that is to do a protein alignment like we've done here, and then we can generate the corresponding DNA alignment using a program called pal-to-nal, which has just recently been added to Birch. And in order to do that, then we have to read the file that contains the DNA sequences that were used to translate the proteins that went into this alignment, and then we run pal-to-nal, and we get a set of aligned DNA sequences. So codon for codon, we now have the corresponding alignment that goes into the amino acid alignment on the left-hand side. So what I want to show you is some of the steps in parallel. We'll go through the protein alignment example first, and then we'll do the same thing for the DNA alignment and see how similar are the two trees that we get when we do phylogenetic analysis on these two data sets. So one of the first problems we encounter, of course, is the heterogeneity of the amino-terminal ends of proteins that is very common among proteins. And so we would need to probably have a way of editing out these very, very gappy regions, because if you think about it, a gap represents probably a single insertion deletion event, but when you have multiple gap characters, that kind of implies each gap character is an independent mutational event, and that's not realistic for the way protein and DNA sequence evolution works. So fortunately, there's a program called Gblocks that does a very good job of editing out the most gappy regions. So I've selected all of the sequences, and I'm going to run Gblocks, and by default it will require that a conserved position be at least 51 present in at least 51% of the sequences, and the flanking positions, what percent of the sequences have to be in a flanking a region flanking a gap. So I tend to set only positions with less than or equal to 50% gaps are included in the output. So let's take a look and run Gblocks, and we get two output windows. One has the output alignment with just the sequences, or I should say the positions that we wanted in the output. So you can see here some gaps are preserved, and it's only the ones that are not terribly drastic. To get a better idea of what was done in the alignment, you can see here that 283 of the positions in the alignment have been preserved. So it actually gives you the alignment, but the great thing is it uses a blue line to underscore those positions that were conserved, or I should say that remain in the final output, and the ones that don't have an underscore are going to be excluded from the output. So you can see this big gappy region is entirely excluded. And so now we have a much more realistic subset of the alignment to work with. So I'm going to skip ahead and assume that we've already run, in this case, maximum parsimony on the protein alignment, doing bootstrapping. So that means creating 100 replicate data sets from the alignment and creating 100 trees from that. So here we see the consensus tree based on those 100 bootstrap replicate trees, and the number at each node represents the number of times each branch was present among the 100 trees that were tested. And so this actually points to one of the issues with phylogeny programs. Is very often you can have a tree that is a consensus tree. It's got bootstrap replicates, but it is not the final tree and doesn't have branch lengths. Or you can have the tree with the branch lengths and not have the replicates. And so then the final thing, of course, is all you see here in the tree is accession numbers. And what you'd like to have is more information like taxonomic information, for example. And so we're going to address all those issues here rather than the mechanics of constructing a phylogenetic tree. So the solution comes from the fact that you can save the different bootstrap replicate trees in a file. So we have it actually in two ways. We have the bootstrap trees in BL3, which is a biolegato for trees, and then we also have those same trees in a text file. And so I'll save the text file for later use, and then we can read in the final tree. And so, for example, here's the final bootstrap tree in BL3. We can use that as input for the maximum likelihood program ProML, which will construct a tree with branch lengths and then we'll add the branch lengths to that tree. So now I'm going back to the Gblocks generated alignment, and I'm going to run ProML, that is the protein maximum likelihood program. And so rather than build a new tree from scratch, I'm going to read in the tree that we built, I should say the consensus tree that we built from bootstrapping. And so I've read that file in, and now I'm going to run ProML. This will be very quick because we're only considering one tree. And so the output now will give us, of course, the tree file this time with branch lengths but no bootstrap values. And so we get that in a couple of ways. I'm actually going to close up Archaeopteryx here and concentrate on the maximum likelihood tree. So we have the tree in BL3 and we have it in text form as well. And in Birch 3.8, we've added a new feature called Conf Add that will add confidence limits, in other words, the bootstrap replicate numbers, to an existing tree. So we just have to find that file, which is the kite3.pro.all trees, and we're going to then run conf add, and conf add will open up the output in a new instance of Archaeopteryx. So there, now we have the, I'll just make the fonts bigger so we can see, and we have the output tree. You can see the bootstrap numbers are there. And then also you can see that the branch lengths are not fixed. The branch lengths, I can even put the branch lengths on if I want. So they're all the branch lengths from the maximum likelihood tree, and we have the bootstrap numbers as well. So even at this point we're not done, because it would be nice to have extra taxonomic information. And Archaeopteryx gives you ways of doing that. Since the names of the sequences are all accession numbers, we can run first of all, obtain sequence information, and you'll see at the top it's running the process to retrieve the taxonomy information from the database that the sequences came from. So I edited out the time it took to run that, and then we'd like to have not just the genus and species information. I guess I can show here we only have genus and species, but I'd like to get the higher taxonomic information. So there's another tool called Obtain Detailed Taxonomic Information, and we're going to run that again as well. So we now have that information, and we're ready to do some additional formatting to make it easier to look at the tree and examine and learn things from the tree. So Archaeopteryx has a whole bunch of tricks that let us improve how we visualize our sequences on a tree, and one of them is coloring by species. It helps you get an idea of how the genes in a gene family cluster together within a species. So one final thing I'm going to do is just make sure that I save the tree, and this will save all of the data in phylogeny, and it's probably worth just having a look at what a phylogeny file looks like. So as I said, this is a very rich format for storing all of the types of information you need regarding a tree. Every clade has a name and a branch length and in this case the taxonomic information now and the bootstrap information. So all of that data is saved there's the bootstrap value there. You can see all of that data is saved in the phylogeny file. So finally, we're at the point where we can compare the protein tree on the left and the DNA tree on the right. And in this particular case, it seems that we got essentially the same tree from both data sets. You can see the woody plants, oak, cannabis, malus, etc., poplar, all clustering separately from the legumes, which are shown in green at the bottom. And so this is a case where the gene families seem to evolve fairly rapidly. Now we do have a case, for example, here in the soybean family, we have one set of genes and then some others which would be different paralogs of the same chitinase 3 gene. And so this is probably more ancient gene duplication that has persisted over time. So while it's true that Birch is a collection of hundreds of bioinformatics programs, the most important point about Birch is how it puts them all together into a coherent, usable package. So for every type of data, there's a BioLegato application that can package together both the data that you work with and the programs to work with each kind of data. So at the end, we have a comprehensive set of bioinformatics tools that all work together and make our lives easier. Thank you for watching today, and if you're interested, you can download Birch for Linux or Mac OS X on the slide that we see here. And please also remember that free bioinformatics software is free, but you pay for it by citing it in your publications. So thank you for watching, and have a look at the other videos on the Birch YouTube channel.